most of the like swine industries are doing the like early weaning between like 3 to 4 weeks of the pig's age. But some like farm also conducts the 15-day weaning or like 16-day or the 18 days of the weanings. So as I mentioned like previously, so they are in the middle of the immature like GI tract development, which may cause the loss of like adverse like effect. So when the early weaning like occurs, so it aligned with the some of the like functions of the GI tract, which will increase the intestinal like permeability, which we can call as the leaky gut. And also it may cause the immune suppressions for sure, because it will increase the intestinal permeability. So they have like much more chance to get invade of the pathogen through the intestine. And also it can be caused the hyperactive like enteric nerve system by the early weaning.
Hello and welcome to the Swine podcast. I'm Dr. Tara Donovan, host, and I'd like to introduce my guest today, Dr. Kwangwook Kim. Dr. Kim is an assistant professor in the Department of Animal Science at Michigan State University. Welcome to our show, Dr. Kim. How are you today?
Good. Thank you for your introductions, and thank you for having me.
Absolutely. So Dr. Kim has been working in the area of intestinal development and nutritional intervention in young pigs. So before we learn about Dr. Kim's research, let's get to know him a little bit better. Dr. Kim, can you give us a quick summary of your career path?
Oh yeah, for sure. So as Dr. Donovan like mentions, so I just like started my like faculty position at the Michigan State University in Department of Animal Science. So it's gonna be my second year as my faculty positions. So before that, so I did the graduate the program at University of California, Davis, including the master and the PhD. So I used the enterotoxin E. coli swine model to testing the different like feed additives to reducing their like adverse effect and also to maximize the productions. So at MSU, my research interested in aligned with my previous like study, which including the evaluating the feed like strategy for like optimal like swine nutritions, more focused on the promotion of the growth and also the health, and also to kind of like achieve the sustainable and the efficient like swine production like overall.
Great. Well, definitely an area that we continue to need to research and learn more about as we see some enterotoxigenic E. coli ebb and flow in our industry for sure. So we're going to talk about intestinal development of young pigs, and then a bit about enterotoxigenic E. coli infection. And then lastly, some nutritional interventions, as you mentioned, that you've been working on. So let's start with just give us a quick review of young piglet intestinal development and what's important about it, kind of the basis of our discussion today.
Yeah, sure. So I would like to start with the gastrointestinal like tract development during the kind of like weaning period. So as we all know, once the piglets get like buried from their like mom, so their like gastrointestinal is like, I'll say like very fragile. So they're not like, exposed to any other like just from the like outer sources, and those like gastrointestinal tract are only related to the maternal immunity from the colostrum and also the milk. So they are not like fully like developed, and they're still immature. So they're not like fully functions to absorbing any kind of like nutrients, and also they're not like quite like functioning for the epithelial barrier to against the pathogen infections, including the immune system like maturations. And also one of the most like hottest like topic in these days is gonna be the gut-brain axis. So they are not still like immature, means they're still immature. So enteric nerve system are not like fully like mature as well. So lots of kind of the immature like gastrointestinal tract like development during the weaning period. So it can be as I mentioned, like fully like rely on the like a maternal like a side. So for the gastrointestinal tract development, so based on like lots of like literatures, it could be like fully like developed as we emphasize it's gonna be the 12 to 14 weeks of their age. But the reason why we are thinking and then we are just focusing on the GI tract development is caused by the early weaning. So most of the like swine industries are doing the like early weaning between like 3 to 4 weeks of the pig's age. But some like farm also conducts the 15-day weaning or like 16-day or the 18 days of the weanings. So as I mentioned like previously, so they are in the middle of the immature like GI tract development, which may cause the loss of like adverse like effect. So when the early weaning like occurs, so it aligns with the some of the like functions of the GI tract, which will increase the intestinal like permeability, which we can call as the leaky gut. And also it may cause the immune suppressions for sure, because it will increase the intestinal permeability. So they have like much more chance to get invade of the pathogen through the intestine. And also it can be caused the hyperactive like enteric nerve system by the early weaning.
Meet AccuFast, your trusted partner in raising efficient, healthy, and sustainable pork. We're not just about genetics, we're about tailored solutions that set you on your path to success, no matter how you define it. At AccuFast, we channel our investments into 3 crucial pillars: our genetic offering, proprietary technology platform, and leading commercial measurement system, ensuring tangible results for our partners. Visit our website at AccuFastSwine.com or reach out to an AccuFast customer success rep to discuss how we can help you create the future you've been working towards. AccuFast, the best way to predict the future is to create it.
Right. So what I heard you say was it's important at this time of age because we're weaning the pig, and the younger we wean the pig, the more permeable the intestine is, or we call, like you mentioned, the leaky gut syndrome. And so then in addition to that, the immune system is just being developed, right, for that pig. And really, the development goes all through the course of what I would call the nursery phase into 14 to 16 weeks of age, which is essentially for a pig, heading towards the middle part of their life with us, right? So they're developing their intestinal immunity really all throughout their life, if in terms of a market pig. So what about the stress of weaning? What does that do? And you talked about wean age, and so the younger the pig, the more permeable the gut. But what about just the stress of going from mother's milk to a diet, a pellet diet in the nursery?
Mm-hmm. Yeah, so there's a lot of like stress like factors can induce the weaning stress, as we call. So not only the like change of the diet from the liquid like milk, sour milk to the solid diet, but also like sometimes pigs need to be transported to the other farms. So pigs needs to be in the like trailer for like 6 to 8 hours to the other farm. So that's going to be another like a stress like factors that can induce the weaning stress. And also when they are like mixed with the other litters. So as we all know, the pigs are the kind of like social like animals. So they are trying to like fight each other to make the like high rank and the ranking and the high rank systems. And also the another like a weaning like a stress can be induced from the as I mentioned, the gastrointestinal like development. So they're very like hard to like absorbing the nutrients. So at the same time, so as they all know that they are trying to like refuse the feed because they're not familiar with the like kind of like a new introduced like feed. So like first like 48 to like up to like 72 hours, they are trying to like refuse to eat. So it may reduce some of the growth performance and also the immune responses. So it may cause the morbidity of the infectious disease and also the mortality.
Right. So weaning is just one stressful day, right,
Mm-hmm.
for that pig. So what about, let's talk about E. coli. What does E. coli do? So enterotoxigenic E. coli is a common pathogen that we see in young piglets. And so what does it do to the gut, and how does it work alongside some of the other stressors?
Yeah, correct. So for the E. coli, it's gonna be the one of the infectious can induce the infectious disease in the pigs. So there are like lots of like different like, types of the E. coli that can induce the some of the different types of like morbidity and then the result in the pigs. But the one of the most like common and then the most like serious like threat for the swine industry, we call as the enterotoxin E. coli. We can abbreviate the ETEC. So ETEC can can be like invade into the gastrointestinal tract and causing the diarrhea, which is kind of the most like important and also the easy to like see from the SEA clinical signs. So there are like two like a major like different like strains, kind of the major strains that can like, induce in the ETAC diarrhea, which include the F4, which we can also call as the K88, or the F18 like strains. So F4 strain can induce the both like pre-weaning and then also the post-weaning diarrhea. But specifically for the F18, they are mostly inducing the post-weaning diarrhea, because of the F18 receptors can be like proliferated in the gastrointestinal tract about like 3 to like 4 weeks of their age. So and also the ETAC is not the only one like a thing or the bacterial infection that can inducing the post-weaning diarrhea. So post-weaning diarrhea that can be happen without any like infectious disease. So as I mentioned like earlier, so the weaning stress itself can increase the intestinal permeability. So it will inducing the leaky gut, and it can also induce the diarrhea. But at the same time, because they have the leaky gut, so they have more chance to other like a pathogen can invade inside of the intestinal tract, including the ETAC and also the virus and also the other like the bacterial disease.
Right. So it also makes the intestine vulnerable, right, for other pathogens to get in. And so you're working on some nutritional interventions, and I know you're studying the interventions looking at an E. coli, a model of an E. coli infection, but there may be some other applications and other pathogens, right, that it also helps with. So let's talk about those. Give us a quick rundown of what you've been doing in the nutritional intervention area.
Okay, so actually, it's just my nutritional intervention was like focused on the non-nutrient like feed additives. So when you just think about the nutritional interventions, so I can classify as like three like different like items, including the macronutrients, including the carbohydrate, protein, and also the fat sources, and also the micronutrients such as the minerals and the vitamins. So many studies have been conducted for the macronutrients using the like different like carbohydrate sources, including the fibers, and in case of the like proteins, so we can also modulate some of the concentration of the amino acid, and also the different like fat sources. So for the macronutrients, we can think about the zinc or the copper or the like high doses of the vitamin C, or either like vitamins or the combination of the vitamin and mineral mix. So those are the two like other options we can think about. So, but I just majorly I just used to work with the non-nutrient like feed additives. So when you think about the non-nutrient like feed additives, so most like common the non-nutrient feed additives, it was the like antibiotics. So antibiotics have been widely used and routinely used for not only to prevent the disease and also to treat the disease, but also it can be used for the increasing the growth like promotion purposes. So, but as we all know, the antibiotics as the kind of growth like promoting purposes have been banned since like 2006 in EU and also the 2017 in the United States. So many like researchers, including myself, are trying to look for other potential antibiotic alternatives like feed additives.
Right, and I like how you categorize those. That's helpful in terms of, sort of three pillars. And so your area of expertise and what you've been working on is non-nutrient feed additives and alternatives to antimicrobials.
Yeah, that's correct.
All right, so tell us what you've been doing, and what are these additives, and what do they do to the gut, and how can we use them out in the field?
Yeah, so today I'd like to introduce and discuss about three different types of the feed additives into the ETAC infected model, and then I'd like to switch gear a little bit for the kind of healthy pigs for the feed additives. So our my objective was the kind of using the antibiotic alternatives for the healthy gut. So it is very hard to identify how to identify and also how we can say gut is healthy. So we have to think about different approaches. So starting with we also have to think about their a major role as the gut, which is the kind of digesting and absorbing the nutrients. And also in case of the immune responses, so they need to be worked as the gut barrier functions, and also they provide the important role for the mucosal immunity, and also at the same time they have to against from the lots of like different like stress factors. So and then finally it can be like modified, and then just they can be set up as the like healthy like gut microbiota. So my research like objective was kind of like combination of the you're looking for the antibiotic alternatives for the healthy gut. So the first like feed additives that I want to introduce is going to be the beta-glucans. So this was kind of my very first like animal trial using the ETAC models. So the beta-glucans can be defined as the group of the polysaccharide, which can be presented into the like a cereal grain or the fungi or like seaweed and also the algae. So they have like different like, types of the beta-glucans. And then it can be also draw like many attentions for the immune anti-inflammatory effect and also the anti-tumors, and also it can also do other like a biological like effect. So we use the beta-glucans for the ETAC infected model using the two different like doses within the low dose and high dose, and then we just identified the beta-glucan was like beneficial to reduce the frequency of the diarrhea when the pigs were infected with the ETAC. And also it will reduce some of the inflammatory effect. So that is why it may reducing the diarrhea overall. And then it can also suppress the pro-inflammatory like cytokines, including the TNF-alpha. And also it shows the reduce the cortisol and the haptoglobin concentrations, which is gonna be the biomarkers for the stress response during the weaning period. And then the one of the most like interesting data was the dactin gene expression in the jejunal and ileal mucosa, which is the part of the small intestines. So dactin is gonna be the one of the receptors for the beta-glucans in the that can be presented into the immune cells, such as the macrophage. So what we understand is that the beta-glucans enhance the expression of the dactin receptors into the immune responsive cell. So that can help to mitigate the ETAC infections in the early weaning pigs. So it will like finally reducing the diarrhea and also the immune responses for the in case of the using the beta-glucans as a feed additives. So this was kind of the like first feed additives that I want to introduce. And then the second feed additive that I want to talk about is the probiotics. So we all know as the probiotics, but when we are using the probiotics in the animal sides, we have to talk as the direct fat microbials. So also I'll use the probiotics in this show, but so like easier to understand. So we use the bacillus like based like probiotics. So bacillus have been identified for the producing the like fiber degrading enzymes, so they can help to improve also the beneficial for the gut microbiota, and also help to like a nutrient like digestion and also the neutralizations. And then the bacillus based probiotics have been known for the easy to handle in the harsh environments. So they can be like we can like store in the farm or as the feed like a long term, and also bacillus based like the probiotics that can like survive in the low pH such as in the stomach. So it can be reached up to small intestine and the large intestine for the like further beneficial effect. So we also use the bacillus based like probiotic to the ETAC infected like pigs. So we also like find out that the probiotics, the bacillus like probiotics increase the feed efficiency in the pigs, and also reducing the like a diarrhea effect. So one of the most important and also the significant result for this is the reducing the permeability in the small intestines. So previously, so I just mentioned about the leaky guts. So it will reduce the leaky gut by the bacillus like based like probiotics. At the same time, they also enhance the like tight junction proteins, which we can between the epithelial cells, so make the sealing the like intestinal tract to prevent the any kind of removal from the intestine to the abdominal, so they can like secure some of the nutrients, and also they can block some of the pathogens. And we also analyze we also like looking for some other kind of like a metabolic pathway due to the bacillus-based like probiotics. So we tested the approaches using the metabolomics and also the 16S rRNA sequencing. And so based on the metabolomics, so bacillus like based like probiotics improve the like protein like synthesis and also the amino acid synthesis. So we were kind of hypothesized that the bacillus like based like probiotics help to enhance the amino acid utilizations in the body. And also when we are looking for the 16S microbial alpha diversity, so it will increase the like diversity in the gastrointestinal tract. So we were kind of like looking for what kind of like a different microdiversity was looking for was the firmicutes and then probiotics, and I mean the proteobacteria. So when we look for the kind of get deep into the family level, so bacillus like based like probiotics reduced to the lactobacilliaceae and increased to Lachinolacspiraceae, so which is responsible for the like energy like harvesting and then preventing some of the pathogen like infections. So those are the two kind of like microbial microbes that can be used for kind of looking for the biomarkers for the ETAC infectious model. So bacillus like based like probiotics help to kind of like modify those two like microbes in the bodies.
I was going to stop you and say that that's a lot of really technical information, and what it sounds like to me is that you're really understanding the mechanism of how these two non-nutrient feed additives work, right? So I'm gonna go back just a little bit just to make sure that I heard you right and to clarify kind of
Okay.
all the technical stuff that you just sent to us. So one was you were understanding how to measure gut health, right? How do you measure these products and if the gut is healthy or not healthy? And so once you determined that in your research and your model, then you were looking at beta-glucans that are polysaccharides, and they decrease the inflammatory effects, right, of ETEC or stress. And you also were able to measure the kind of stress effects of that weaning period, and using beta-glucans, it helped with the cortisol and haptoglobin levels that were measured, right, to give you an idea that's helping the stress on the gut. So did I hear that right? Did I get that right for a quick summary?
Yeah. So
very basic.
I'm sorry, yeah. So I did. I think I just did like to like dip in for the technical like measurements. So do you want me to just go over one more time for the kind of like briefly talk about the beta-glucans and then the probiotics?
Yeah, well, I think we heard you. I'm just making sure that I heard you right in terms of two different kind of products or non-nutrient feed additives that you're working on, beta-glucans and then probiotics. And what I'm understanding is that you understand the mechanism at which those two additives help the gut, right, in response to these stressors. And so those mechanisms are helpful for us to understand, so that we can time the quantities and the period of time when we feed these types of products to the pigs, and so we get their maximum benefit.
Yeah.
Okay. Well, good. So let's go back to you were talking about the probiotics and how you're able to measure the bacillus and the probiotic and what it does to the intestine. And you're using some measurements of the other, what I call good bacteria, right, that are in the gut that say, yep, this bacillus is helping those good bacteria to live and grow. And so expand upon that just a little bit and what you found. How do you sort out there's some different bacillus strains, right, that you can feed. So is that how you're determining which ones to add into the feed of the pig?
Yeah, so the bacillus like base, so there are like numerous like literature have been used for the different like types of the bacillus based probiotics. So we especially use the bacillus subtilis. So it has been used for the increasing the fiber degrading enzyme, and also it may help to kind of nutrient like digestion and utilizations by the modifying the like good like beneficial like bacteria in the gastrointestinal tract. So we use the bacillus like subtilis for the E-tank model, and then we found out that the bacillus like subtilis enhance the like overall like daily gain and also the feed efficiency of the pigs with infected with the pigs. And also one of the notable like result for the bacillus subtilis was the reducing the intestinal permeability, which is kind of preventing the leaky gut from the pigs infected with the E-tank. So they can prevent some of the losing the nutrient from the gastrointestinal tract, and also reducing the diarrhea. So, and also when we think about the what kind of like metabolism and also the metabolic pathway underlying the bacillus like subtilis, so it was like aligned with our like hypothesis, which is that the bacillus subtilis were like help to like aiding the amino acid metabolism, and also it modified some of the like bacteria in the gastrointestinal tract to reducing the infectious like disease caused by the E-tank, and also it will has some of the more like bacteria, including for the promoting the higher like energy harvesting that can be helped to increase the growth performance.
So if that product, the probiotics, help with a leaky gut syndrome, I would assume that a good procedure then is to try to get some of those products fed before weaning to try to, maybe do a creep feeding. And so that's I know an area of focus out in the field is, when's the right time to creep feed? What should your creep feed have in it? And so it makes sense to me that if that really helps in that leaky gut, and you're looking at a wean age, right, that the lower the wean age, the more leaky the gut, that would be a good strategy.
Yeah, I think that's a great point. So when we introducing the kind of like different types of the probiotics, I think as you mentioned, the earlier could be better, because as I mentioned for the first like part of the gastrointestinal development, so they are like still like kind of immature of the gastrointestinal like functions, but at the same time, but they're not like their like microbial like diversity are not like fully like set up. So when we are introducing more like diverse like beneficial like probiotics in the creep feed, and also the early time of the weaning period, that could be like beneficial for the pig health.
Yeah, exactly. Good. So we're kind of ending our time. Is there anything else you want to talk about? Any additional things that you've been working on, and maybe just a quick summary for our group on our conversation?
Yeah, so as I just introduced about the E-tag infected like pigs from my previous study, and then I'm trying to do some other like stuff at MSU, and then I'm testing the kind of different like feed additives, not for the E-tag infected pig, but also the kind of like commercial like base like pigs in like kind of we call it as a healthy pigs. So we recently tested the non-nutrient like sweeteners. So we can also call as the artificial sweeteners or the sugar like substitute. So I believe like there are like lots of still like debates on about the like the sweeteners. So what we found out is that the the non-nutrient like sweeteners that we tested has the positive effect on the growth performance and reducing the diarrhea. So we are very excited to looking for more like underlying mechanism. So how those like, kind of like sweeteners are using into that. So I'm very excited for not only for the sweetener and also for the other like future like research using the feed additives for exploring the antibiotic alternatives.
Right. No, that sounds interesting. And so I like that you're understanding the mechanisms and helping to teach me how these products actually work inside the pig and why they're beneficial, because, there's a cost to them, right? And trying to get them timed right and the amount that we're feeding correct. Understanding the mechanism helps us to determine that a little bit better, to fine tune it.
Mm-hmm. It's time for our famous 3. Feed flow is changing the way the swine industry sees feed. As the world's only on-pipe feed sensor, FeedFlow tells you exactly how much feed is being delivered to your animals, so you can be sure that every pig in your barn is well fed and growing. With industry-leading precision and up-to-the-minute real-time data and alerts, FeedFlow is a simple and affordable way to improve production outcomes across your organization. Feed is too expensive to ignore. Try FeedFlow today.
All right. Well, we're going to ask you the three questions that we ask all of our guests. And so one of those is, what is your favorite book or resource that you use in your area of expertise? So what's your go-to for pig health and immunity?
Okay, so I think there's like a book right next to me. So I just like started my like study using like this book, which is the Swine Nutrition. So this is kind of, I'll say like kind of like a Bible for the swine nutrition, and then they have like lots of like information talking about the different like nutrients, and then not only for the nutrients but also the most of the important like stuff for the swine like management, and also So I think I recommend the Swine Nutrition for the my go-to book. All
right. So how about outside of swine nutrition immunology? What's something that you've read recently, or a website you've gotten, or a conference you've gone to recently that you think those of us out here that are also in the industry would find interesting?
So as a like swine nutritionist, I really enjoyed the conference we call as the ASAS Midwest meeting. So there are lots of like swine nutritionists, and then just introducing their like research, and then talking about the abstract, and also providing some of the great talks. Then also I really enjoyed the time in the ASAS Midwest meeting in March.
All right, so not long ago. So that's a big conference that I know a lot of really good research like what you've been doing is presented with a lot of students as well, which is great. Right. To have some student competitions, posters, and presentations. Great. And so lastly, share with us an attribute or characteristic that you feel sets successful professionals apart from others.
Well, that's the hard questions to answer. So I think it is very important to kind of like communicate with others. So I think it is very like easy to like get to like isolated by yourself, because when you are doing your research by yourself. So as I mentioned like earlier for the conference, I think it's very important to like communicate with the others. So presenting your like a data, and also like discuss with others, and then get some like advice, and also looking for other just technical like a measurement or method, or other kind of a different like a way to improve your like overall like career, I believe. I think that's the most important thing.
Absolutely. And, that's the whole basis of our Swine It podcast show, right? Is to get people like yourself, Dr. Kim, who are out there doing a lot of really good research on pathogens and mitigations that we can use in the field. But how do we get them, how do we get you out into meeting up with veterinarians who are dealing with E. coli out in the field? And so this is exactly the way to do that. So I agree, communication is really key to getting the information out there and all of us learning and improving.
Yeah, correct.
Well, it's been a joy to have you on the show. I appreciate your time. If you have questions for Dr. Kim, post those on your platform on the Swine It podcast show. And thanks so much.
Thank you so much. Thank you for having me again.
Absolutely.